Thank you for joining me today to discuss the capabilities of our critical reagent program here at Eurofins DiscoverX. My name is Neil Charter and I am the Head of Operations and oversee the activities of our cell production group here at our site in Fremont, California. Analytical bioassays for biopharmaceuticals are required to meet critical requirements in order to be used for lot release. These bioassays must allow for an accurate measurement of relative potency of the drug and reflect the drug's appropriate mechanism of action. Given that the lifespan of a biologic can be up to 20 years, it is essential that the assay can be supported for the long term. Therefore, it is crucial that production bioassay lots are reliably manufactured from cell banks that have been fully characterized and provide sufficient redundancy to mitigate risk. In addition to functional performance, other parameters need to be considered when qualifying cell banks as critical reagents, such as purity, sterility, passage stability, as well as enhanced storage environments for the long-term use. In this presentation, I will describe the strategy used to generate critical reagent cell banks to support multi-decade production of Eurofins DiscoverX bioassays. Reliable bioassays need to be generated from a well-qualified stock and be designed to fit the needs for potency release assays. Source material qualification is important because it ensures that the cell stock that are used to produce the bioassay have a traceable history, are free from contamination, and stably express the target of interest. The generation of a two-tiered cell bank provides an effective way to guarantee that bioassay stocks can be manufactured reproducibly by providing a well-characterized stock with a defined and limited passage number. Performance qualification is critical to establish that the assay is fit for purpose by demonstrating the accurate potency determination, specificity, dynamic range, and precision. Combining these factors ensure that the assay is robust and effectively measures small changes in biological activity of the therapeutic. Quality in production is key to providing lot-to-lot consistency so that reproducible results can be obtained throughout the lifetime of the drug. Having a defined strategy for critical cell bank reagents is an essential component in providing long-term support in the QC lot release program. We utilize a multi-tiered approach to ensure that the bioassay production lots are manufactured in a reliable and defined manner. Primary and source clone banks provide a fully characterized platform for critical reagent banking. Two-tiered banks consisting of a master and working cell bank enable the cells to be scaled up using a limited number of passages. Each vial of a master cell bank can be used to propagate a new working cell bank, which in turn is used to manufacture production bioassay lots. With this strategy, we are able to deliver long-term supply of bioassay kits with excellent redundancy and risk mitigation. Initial clone stocks have a limited number of vials, and so must be expanded in order to create a bank that can be used for both continuous culture and a source for master cell bank generation. The clone is tested for stability for up to 50 passages to confirm that the cell line does not deteriorate over time and can perform over a number of passages needed to generate both master and working cell banks plus the bioassay production bank. Each stage needs to be appropriately qualified to verify that the performance and the quality of the clone is maintained. Dual site storage is employed for the primary and master cell bank to mitigate any risks at the main storage site. Source and master cell banks undergo extensive testing to confirm the identity of the cell background and that the stocks are not contaminated with either other mammalian cell types or bacterial, viral, or mycoplasma pathogens. To qualify the source, understanding the lineage of the progenitor cell line is important. For that reason, we verify that the source of our cell lines are derived from ATCC stocks. The cell stock is engineered with our proprietary technology to report out an accurate mechanism of action for the drug therapeutic effect. Stocks are tested for a battery of pathogens, including key viruses, bacteria, and mycoplasma species, and it is established that the cells are not contaminated with other mammalian cell types. Our source cell lines are extensively characterized to demonstrate their suitability for bioassay development. Alongside confirming extended passage stability using the assay functional readout, the cells are tested for surface expression in parallel using specific antibodies. This data provides an excellent verification of the correct expression and localization of the targets of interest. Cell culture parameters such as morphology, optimal growth conditions, and defined doubling rates are established to aid in bank generation and provide a reference during propagation. The source cell stock is tested for purity, which confirm that the cells belong to the correct species, that there are no contaminating cells from different species, and that the cell line marker profile matches that for the original cell background from ATCC. Finally, the stock is confirmed mycoplasma and bioburden-free using compendial methods established by the US and European pharmacopoeia bodies. Combined, this data set confirms that the source clone has the correct lineage, expresses the target of interest in the correct format, and is free of contamination. Quality during cell production is driven by defined raw materials, established procedures, detailed documentation, and oversight from quality assurance. High quality materials are important, especially in cell production, and to that end, premium serum is used that has full documentation on the source. This ensures that the cell stocks are cultured and cryopreserved using the best components available to meet regulations in all major geographical locations. Manufacturing is defined and follows standard operating procedures that are fully controlled by quality assurance. Cell production is documented in a traceable manner and captures high-level details about the health and performance of the cells at every stage in the process. Documentation is managed in a production database which automates the calculation of production metrics to ensure consistency and accuracy. After cryopreservation, the cell stocks are put through a rigorous battery of tests to confirm the functional performance, cell viability, and mycoplasma and bioburden-free. Defined acceptance criteria are used for each metric, and we leverage the capabilities of our production database to automatically determine if the QC results meets the specifications for the product. The results are then reviewed in a two-tier process to confirm that the stock passes QC and all documentation is complete. Quality assurance performs an independent review of production and QC data to qualify the lot for release. CFAs are automatically generated from the qualified production data and electronically signed off by the production manager and QA. Production quality is controlled using a broad set of QC tests to ensure optimal bioassay performance. In a critical reagent program such as this, establishing that the master and working cells are of high quality is essential since these are needed to produce bioassay stocks for up to 20 years. Functional performance, as shown here, is tested on vials at the beginning, middle, and end of the freezing process, so that vial-to-vial reproducibility within the lot can be determined. Qualified reference lots are tested in parallel to ensure that there is lot-to-lot reproducibility as well. Intra and inter-lot variability is calculated as a percentage relative standard deviation and must meet or exceed the acceptance criteria established for both assay potency and assay window. As shown here, the master and working cell banks produce exceptional vial-to-vial and lot-to-lot reproducibility and result in the same level of performance as the corresponding bioassay lot. Additional factors such as viability of fall and cell recovery examine the robustness of the frozen cells as they are being revived and at the time of the assay. Cell purity is also established to ensure that the stocks are free of mycoplasma and are bioburden contamination. Together, the QC requirements provide the evidence to demonstrate that the critical reagent cell banks are qualified for production of bioassay cells suitable for QC lot release applications. In addition, master cell banks are tested every 4 years to confirm that the stock performance continues to meet the requirements over the lifetime of the program. Long-term support for QC lot release programs relies on good lot-to-lot reproducibility to provide consistency in potency determination over time. The control charts here demonstrate that our analytical bioassays exhibit excellent lot-to-lot consistency over the span of 15 lots and up to 5 years of data. No significant trends in assay drift have been observed, and the mean potency values for the reference molecules are less than 2 standard deviations of the historical average. Average reproducibility within a lot is within 18% relative standard deviation. This data demonstrates that these analytical bioassays can offer consistency in performance over multiple years, which is required for QC lot release programs. Documentation is provided on our working and master cell banks. These give details on the cell line characteristics, including growth rate, passage stability, morphology, and receptor expression profile. Manufacturing information includes freeze date, vial density, passage number, and requalification date. The quality control section provides a summary of all the tests conducted to qualify the lot. That includes the functional results, viability metrics, and identity testing. For the biological safety, the results of mycoplasma bioburden and pathogen testing are also documented. The CofA is verified and signed off by both the production group manager and quality assurance. Our manufacturing capability is predicated on an organized facility to ensure continuity, capacity, quality, and a guaranteed supply of critical reagents. All of our raw materials are sourced or manufactured by DiscoverX, allowing us to control the quality of the reagents that make up the bioassay kit. Two-tiered cell banks provide the platform to generate reproducible bioassay production lots for the lifetime of a biologic therapeutic. We utilize automation in many areas of cell production, particularly for vial labeling, filling, and capping. This eliminates variability, especially for large lots, and improves the cell quality by reducing packaging time before cryopreservation. Bioassays are optimized for performance and reproducibility. Assay precision and accuracy are determined, and suitability for relative potency demonstrated during qualification. With master and working cell banks in place and bioassay lots available for immediate evaluation, this provides an optimal platform for implementation in a QC lot release program. To give greater usability for bioassay kits, we offer excellent shelf life for our assay-ready cell products. Our bioassays have a minimum 3-year expiration from the date of manufacture, and we have been extending the shelf life for many bioassay classes in order to provide even greater flexibility. When a client wants to maximize this shelf life and lot size, they can commission a dedicated lot. This extends both the useful life of a lot and reduces the number of times lot bridging has to be performed over time. Kit stability encompasses multiple aspects. The other components in the bioassay kit have great reagent stability, and as with the cell component, have excellent lot-to-lot reproducibility. Our critical reagent program is designed to provide reliable long-term supply. We understand the need to have consistency for analytical bioassays and verify our critical reagent cell banks every 4 years so they can support the therapeutic life cycle. We manage supply continuity by providing long-term support, controlling the quality of our critical reagents, reducing risk, and ensuring consistency throughout our organization. Product support encompasses cell banking services, reagent storage, full-blown tech support, including on-site training, and a CRO accreditation program. The critical reagent program controls the quality of key materials and cell banks and ensures that long-term supply for third-party reagents. Two-tiered cell banking ensures that we can support drug programs well in excess of their typical lifespan, and off-site storage provides the continuity even if the main site is unavailable for production. Quality assurance is critical to ensure that products are manufactured to the levels needed. All processes operate under our quality management system, and we provide all the support for both client and CRO. All our products are manufactured in the California Bay Area at our site located in Fremont. We utilize state-of-the-art equipment to automate as many of the processes as possible during cell production and cryopreservation. Our facilities have been optimized using lean principles with the focus on delivering fast turnaround with uncompromising quality. Our cell banks are housed in dedicated -150 freezers to provide optimal long-term storage for these critical reagents. With our expert distribution system, we can provide customizable options for shipping throughout the world. Our products are supported by our quality management systems to ensure compliance to our established processes. All SOPs are managed using an industry-standard document management system that is in use by many Eurofins organizations, including GMP and GLP facilities. Training is managed in the system and tracks all pertinent data for our employees. Quality is stressed throughout the organization, and our management take an active role in ensuring that the processes are followed. A continual improvement program, including internal audits and an active CAPA system, is designed to help us maintain our product quality and improve our processes as needed. We believe at Eurofins DiscoverX that expertise, facilities, and organization help us build high-quality products. Our production team are highly experienced and draw from expertise in industrial and GMP environments. We have a strong innovation group with a core competency of assay design who have built 95% of the technology in-house. This provides an edge over the competition since we understand the underlying principles of our technology and can translate that into robust products with superior performance. We utilize many systems and tools to automate many of the processes both in data management and production, which are fully supported by our quality systems. The critical reagent cell banks that we have developed at Eurofins DiscoverX have been extensively characterized and QA approved to provide a solid foundation for the manufacture of bioassays for lot release implementation. Using a two-tiered analytical bank approach is a proven system to provide dedicated cell banks for bioassay supply. Two-site storage ensures that critical reagents can deliver a continuous supply of bioassay cells for the lifetime of your therapeutic program. We offer the capability to generate dedicated cell banks for your molecule with a variety of options for characterization and configuration to suit your exacting needs. To facilitate and smooth transition of a bioassay to a CRO, we offer a CRO certification program. This premier service provides on-site theoretical and wet lab training and allows the organization to gain valuable insight into how to get the most out of our bioassays. Accreditation enables the CRO to be marketed to clients who are looking for partners to support their therapeutic program. Contact us at CRO_certification@discoverx.com for further details. I want to thank you for taking the time to review our critical reagent program. This has been designed to act as a foundation for qualified and MOA-based bioassays. Key to the strategy is a two-tiered bank approach that ensures multi-decade supply of bioassay production. The quality of the banks are verified using a combination of functional performance, independent expression profile, stability testing, and purity confirmation. The result is a robust platform for manufacture of high-quality bioassay kits that provide MOA reflective results with excellent reproducibility and precision. This program is a further extension of our product support for our bioassay kits in conjunction with hands-on tech transfer and specialized supply agreements. Please visit us at discoverx.com/cellbanks for more information. Thank you.